Hi everyone, my name is Surinder Kaur and today me and my partner Jay Patel, we are going to represent our project on the topic Comparative Evaluation of Antibacterial Efficacy of Different Toothpaste Brands Against the Dental Pathogen. Let's start with some background. Dental caries or tooth decay is a major health issue often caused by bacterial biofilm on our teeth. Key pathogens involved include Streptococcus mutans, Staphylococcus epidermidis, Lactobacillus acidophilus, and some species of Klebsiella. Historically, dental caries were less common before the Industrial Revolution when the sugar consumption was lower. Today, the rise in the caries is linked to increased sugar intake and then the need for effective antimicrobial toothpaste. Our study aims to fill a gap in research by comparing the antibacterial effectiveness of four toothpaste brands, namely Close-Up, Oral-B, Colgate Neem, and Colgate Mint, against the five key dental pathogens. We have four main objectives. First one is to evaluate the antibacterial activity of each toothpaste. Second one is to compare their effectiveness at different concentrations. Third one is to determine the mean zone of inhibition for each brand. And lastly, to compare the efficacy of toothpaste to broad spectrum antibiotics. We based our study on three hypotheses. Toothpaste brands vary in their antibacterial effectiveness. Second one is the higher the concentration of toothpaste result in the higher inhibition zone. Third one is toothpaste brand with higher antimicrobial components will be more effective. For our experiment, we used four brands of labeled toothpaste such as A, B, C, and D along with standard test organism and media. We prepared toothpaste solution at different concentration, mainly 2 gram in 5 ml and second concentration is 4 gram of toothpaste in 5 ml sterile water, and then inoculated agar plates with the pathogen, and then we added the toothpaste solution to the wells, and lastly we measured the inhibition zone after incubation. So now my lab partner Jay is going to explain the results and the leftover portion of our PPT. Thank you.
From the study, brandy came out on top with the biggest antibacterial effect, averaging 18.65 mn in inhibition zone. Brand A was a close second at 18.45 mm, and brands B and C didn't do as well. With their inhibition zones at 17.05 mm and 15.90 mm respectively. This shows that different toothpaste brands work differently when it comes to fighting bacteria. These graphs display the antibacterial efficiency of different toothpaste brands. Each graph represents the inhibition zone measured for brands A, B, C, and D. They illustrate the varying levels of effectiveness among the brands. Furthermore, the study also evaluated the efficiency of broad-spectrum antibiotics alongside the toothpaste brands. Chloramphenicol demonstrated the highest efficiency with a mean inhibition zone at 35.6 mm, significantly outperforming all the toothpaste brands tested. Tetracycline showed moderate effectiveness with a mean inhibition zone of 19.4 mm, while ampicillin had the lowest efficiency along the antibiotics with a mean inhibition zone of just 4.4 mm. These results highlight the superior antibacterial properties of chloramphenicol compared to both the toothpaste brands and the other antibiotics tested. So in the discussion, The results of our study substantiate both of our proposed hypotheses. Firstly, our findings clearly demonstrate that there is significant variation in antibacterial efficiency among the different toothpaste brands tested. Specifically, brand D showed the highest level of antibacterial activity, outperforming all other brands. This result suggests that brand D contains particularly effective antibacterial agents which are successful in inhibiting the growth of dental pathogens. On the other hand, toothpaste brands with lower antibacterial efficiency may not provide adequate protection against these pathogens. Secondly, the study supposed the hypothesis that higher concentration of toothpaste generally lead to larger inhibition zones. This implies that the amount of toothpaste used plays a critical role in its effectiveness against bacterial growth. Our data indicates a direct relationship between concentration and antibacterial activity, highlighting the importance of using a sufficient quantity of for optimal results. Despite these promising results with some toothpaste brands, it is important to note that antibiotics such as chloramphenicol demonstrated superior antibacterial potency compared to any of the toothpaste tested. This reinforced the notion that while some toothpaste can provide a significant level of protection, antibiotics are still more effective for controlling bacterial infections. Understanding their continued importance in oral and overall health. So the study concludes that toothpaste brand containing specific antibacterial agents are more effective in controlling dental pathogens, while all tested brands exhibited some level of efficiency. There were notable differences in performance, with certain brands showing significantly better results. This underscores the importance of selecting toothpaste which proven antibacterial properties, as not all products offer the same level of protection. Additionally, our findings suggest that consumers should consider toothpaste brands that provide clear evidence of their antibacterial effectiveness when making their choice. For future research, it is recommended that to investigate the specific active components responsible for the effectiveness of this toothpaste and their potential clinical implications. Further studies could also explore the long-term effects of using this toothpaste on oral health and their interaction with other oral hygiene products, such as such results could contribute to the development of improved oral health products and more informed recommendations for consumers and healthcare um, providers. And lastly, for the presentation defense, be prepared to discuss the rationale behind selecting specific taste organisms and toothpaste brands, the interpretation of inhibition zones, and the implications of the results. This presentation will showcase compare comprehensive understanding of the research and its relevance to dental health. And these are the references for this study. Thank you so much.